Chapter 7 is titled Cellular Respiration. And I know most of us probably think of breathing when we think about respiration. But from a biochemical standpoint, respiration means the use of an electron transport chain to generate energy. So we're going to be looking at how cells carry out their metabolism, and in respiration, the final step is going to involve an electron transport chain. We will literally move electrons from one molecule to the next until we get to the end. That's going to be generating energy for the cell. The first slide here shows these two chemicals, NAD+ and NADH. We will be needing to follow oxidation and reduction reactions. And the device I use to remember that is oil rig, because oxidation is the loss or the removal of electrons and reduction is the gain. In a non-organic chemistry class, when you looked at oxidation and reduction reactions, you were literally following the flow of electrons. With biological reactions, it's still the flow of electrons, but in nearly all cases, that flow of electrons is accompanied by the flow of protons. So an electron is abbreviated E- because an electron is a particle with a single negative charge. And a proton is H+, that's a hydrogen ion. And one electron plus one proton is the equivalent of a hydrogen atom. So for the reactions we are going to follow to determine if it is an oxidation or a reduction, we will look to see if a molecule has lost a hydrogen, in which case it has been oxidized, or gained a hydrogen, in which case it has been reduced. So going from NAD+ to NADH, we have gained a hydrogen. Reduction is the gain of hydrogens. And NAD+ stands for nicotinamide adenine dinucleotide. And that is why it has an abbreviation. You will not need to write out nicotinamide adenine dinucleotide. You will just need to recognize that NAD+ is the abbreviation for that. And the role of an NAD+ molecule is to carry electrons. Meaning, where there are other reactions in a cell that result in an oxidation, the loss or removal of hydrogens, the oxidized form of the electron carrier wants to gain those electrons. So these NAD+, and we will see another coenzyme called FAD, for flavine adenine dinucleotide, they act alongside the enzymes to capture the hydrogens that get removed during oxidation reactions. And then, because they are carrying those hydrogens, that is potential chemical energy. That will be a molecule that we will be keeping track of as we move through the various types of metabolic reactions. You have seen ATP before, adenosine triphosphate. There is ribose. This is the exact same sugar that is found in the nucleic acid RNA. This hydrocarbon and nitrogen ringed structure is the adenine. That is the same adenine that is on a nucleotide. And then we have three phosphates, the alpha, beta, and gamma. And if we break these bonds between the phosphates by hydrolysis, that releases energy to do work in our cells. And if we lose one, we lose the gamma phosphate, we're left with adenosine diphosphate. If we lose the beta and the gamma, we're left with adenosine monophosphate. So adenosine diphosphate is ADP, adenosine monophosphate is AMP. We recycle between our ATP and our adenosine diphosphate plus phosphate We have chemical pools of both of these in our cells, and if we have plenty of ATP, we can hydrolyze a phosphate off. That releases energy to do work. If we have more ADP and phosphate, we can use that energy to add the phosphate back on and regenerate our ATP. We are also going to see phosphates going on to molecules other than ADP. This green molecule, whatever it is, can have a reaction where a phosphate is removed from ATP and gets added to it, and that is a phosphorylation. We add a functional group. We will also in later chapters see dephosphorylation reactions where we remove a phosphate group. There is my poorly hand-drawn phosphate functional group. The mitochondrion we saw in our cellular structure chapters. It is about the size of a bacterium. It does have two membranes. The outer membrane is smooth. The inner membrane has those folds. Those folds are called the cristae. The interior of the mitochondrion is called the matrix. That's the fluid inside the mitochondrion that is analogous to the cytoplasm of the cell. There is a space between the two membranes, and there are enzymes on that inner membrane called the Atp Synthase. That is where our electron transport chain is located in eukaryotes. In a prokaryote, there's only one membrane, the actual plasma membrane. Eukaryotes, the cellular respiration is going to be happening on the inner membrane of the mitochondrion. There is a lot going on this slide. This image and the other metabolic pathway images will be provided on exam 2. This chapter 7 is not going to be part of exam 1, but for the next exam, this image will be present. All of my scribbles on it won't, but the image will be. This is the first half of glycolysis. We've seen lysis before, meaning to break. We've seen glyco or gluco before, meaning sugar. So this pathway breaks sugar. Specifically, it is breaking glucose. We are using glucose as our model food for these metabolic reactions because a large number of living things on Earth can use glucose, and they can use it in this exact pathway. The exact structure of their enzymes might vary, but the way in which glycolysis is carried out really doesn't change between us or E. coli, or a whale or our dog. Anything that's doing glycolysis is doing it this way. We start out with glucose. It's a hexose, a 6-carbon sugar. The first part of this pathway involves inputting some energy. We have to add an ATP here, and we add one here. The phosphates and ADPs are color-coded in red, so you can see where they're being added and where they flow. We've got some greens that are some carbons that are highlighted as green, and some of them are blue, so we can see what happens to them in the pathway. So the first enzyme is called a hexokinase. Hexo because it acts on a 6-carbon molecule. And kinase are enzymes that are involved with phosphate transfers. So it's an enzyme, kinase, phosphate transfers. Why not phosphatase? They didn't ask me when they named this. So the hexokinase catalyzes taking the phosphate off of ATP, and we stick it on the 6th carbon of the glucose. And now we have a linear molecule as opposed to a ring. These carbons here that were green are at the top of the molecule. The carbons that are blue are at the bottom. And this is called glucose 6-phosphate because it is glucose and the phosphate is on carbon number 6. Then we have an enzyme called a phosphoglucose isomerase. So an Isomerase is an enzyme that catalyzes the formation of an isomer. We're not going to add or subtract any functional groups, but we're going to rearrange the functional group. And that's with this blue arrow. We had an aldehyde sugar, an aldehyde functional group on the first carbon with glucose 6-phosphate. When we rearrange this as an isomer, we move the carbonyl from being an aldehyde to being a ketone, and when we do that, it becomes fructose 6-phosphate. Moving that carbonyl from the terminal end to the middle now makes this end, with hydrogens and a hydroxyl, more reactive. Our second ATP comes in, and the phosphofructokinase removes a phosphate from ATP and adds it to the first carbon. So now we have fructose 1,6 biphosphate. I've also seen it called bisphosphate. I've also seen it called diphosphate. All of those mean there are 2 phosphates, and they are on the first and the 6th carbon. So at this point, we've added energy in, and we've done some rearranging. We haven't extracted energy out. The next step, the fructose bisphosphate aldose. There's a mouthful. You're also not going to need to memorize these enzymes. They'll be on the images. This is where we get the lysis. We're going to break the 6-carbon sugar into 2 3-carbon sugars, each of which has a phosphate. We initially create 2 different molecules. We get dihydroxyacetone phosphate, and we get glyceraldehyde 3-phosphate, which is abbreviated G3P. These arrows going back and forth indicate we have another isomerase, which means these are isomers, structural isomers of each other, and the cell will convert the dihydroxyacetone phosphate into G3P, so that we don't have 2 different molecules. We're going to have 2 of the glyceraldehyde 3-phosphates to continue. So we'll have 2 of these that will continue for the second half. So what we've done so far is put in energy and cracked the glucose. The second half of glycolysis will be dedicated to extracting energy. We have to have 2 glyceraldehyde 3-phosphates. That's why I've put 2's all over this image. So we have 2 of these. And now we're going to have 2 NAD+, our oxidized form of the coenzyme, and 2 phosphates that are going to go in. The phosphates get added to the number 1 carbon. This molecule also gets oxidized. We could tell that because we could count up the hydrogens, and there will be fewer hydrogens here. We could also tell that that happened because this molecule at the end got reduced. It gained hydrogens. So if you see one of the carriers get reduced, that means the molecule before it got oxidized. So we have 1, 2, 3, 4, 5 hydrogens here, and we have 1, 2, 3, 4. We lost. We removed a hydrogen from glyceraldehyde 3-phosphate, and we put it onto NAD+. We generated the reduced coenzyme NADH. That is our one form of chemical potential energy. Now we have 2 of this molecule that we're going to have another kinase, and in this case we are going to take the phosphate off of each of these and put it onto ATP, so we generate 2 ATPs. Then we're going to have a rearrangement of our phosphate group from the middle to the end. We remove water, and we end up with a chemical called phosphoenol pyruvate. And our final step is to take the phosphate off of this molecule, put it onto ADP, and make ATP, and we end up with 2 molecules of pyruvate. We put in one 6-carbon molecule called glucose, we end up with 2 3-carbon molecules called pyruvate. During this second half, we have a stage here where we generate 2 ATP, and here we generate 2 ATP, which means we made 4. However, we spent 2 at the beginning, so the cell is only energetically ahead, a net gain of 2 ATP. So we get 2 NADH potential energy molecules. We have 4 ATPs, but a net gain of 2. And that just gets us to the pyruvate. We have to continue oxidizing these carbons, removing the hydrogens in order to complete cellular respiration. So we get from our pyruvate in what's called the bridge or transition step. We're going to take both pyruvates and carry out a decarboxylation. The carboxyl group is removed. There's our carboxyl. To decarboxylate, we remove that. It comes off, highlighted blue here, highlighted blue here, as carbon dioxide. So we have 2 of those. 2 carboxyl groups are removed. They are released as 2 carbon dioxides. This CoASH is coenzyme A, and it has a sulfhydryl functional group on it. It's a coenzyme. It doesn't participate in the reaction other than attaching temporarily to this molecule to make it more reactive for the next step. So it covalently binds temporarily. The very next step, the coenzyme A is going to be removed. So we had 2 pyruvates, so 2 coenzyme A's have to come in. 2 carbon dioxides are released, and we once again have 2 coenzymes, NAD+, that get reduced to 2 NADH. So if we are reducing the NAD+, it has to get its hydrogens from somewhere, and it gets them from the oxidation of the pyruvate. And we end up with acetyl coenzyme A. And the prefix acetyl means a 2-carbon molecule. So we remove the carbon dioxides, we reduce the NAD+, and we end up with a 2-carbon molecule that has the coenzyme A attached to it. So energetically, we have 2 NADH. We don't get any ATP from this. And our carbon still is holding on to some hydrogens. This is the citric acid cycle. It's also called the Krebs cycle. It's also called the TCA cycle. Depends on who you ask. The citric acid cycle tells you what it is biochemically. The first intermediate is citrate, which is also called citric acid. So that is why it was called the citric acid cycle. It was the first intermediate they were able to isolate. Krebs cycle because Dr. Krebs figured all this out. And finally, TCA cycle stands for tricarboxylic acid. And that is because most of these intermediates have 3 carboxyl groups on them. 1, 2, 3, 1, 2, 3. So whichever term you use, I will try to be consistent and say citric acid cycle. I might slip up because I'm also teaching micro and their book calls it the Krebs cycle. But this is one cycle of the citric acid cycle. So we have to double everything to get to our total because 2 acetyl-CoA came out of that transition step. So we have to put 2 acetyl-CoAs in. But if we follow 1, the acetyl coenzyme A comes in and it attaches to this chemical oxaloacetate. It's called the citric acid cycle because there are things coming in and out of the cycle. However, at the end, we regenerate this 4-carbon molecule oxaloacetate that is present for the acetyl-CoA to come in and bind, start the process over. So when the 2-carbon, and that's what this stands for, C2, binds to the 4 carbons of oxaloacetate, we put in water and we remove the coenzyme A. And now we have citrate. From citrate to isocitrate, those are structural isomers. We're moving where the hydroxyl group is. In the next step, we have a couple of things happening. Carbon dioxide comes out, so we're going to remove one of those carboxyl groups. And we carry out an oxidation because our electron carrier, NAD+, gets reduced to NADH. So we had 3 carboxyl groups here. We have 2 here. The other one has been released. It leaves the pathway as carbon dioxide. Going from alpha ketoglutarate to succinyl-CoA, we bring in coenzyme A again to help it be reactive. Going from NAD+ to NADH says we are going to oxidize alpha ketoglutarate to succinyl-CoA, and we release another carbon dioxide. So we had 3 carboxyl groups, we removed a carbon dioxide, and now we have 2. We have 2 carboxyl groups, we remove a carbon dioxide, and now we only have 1. At the bottom of this circle is where we are generating energy. And it shows a GDP, standing for guanosine diphosphate, and a phosphate, making GTP, guanosine triphosphate. Energetically, this is the equivalent to ATP, and the guanosine will be replaced with an adenine, and it will make ATP. When we do that, then we end up with succinate. We have a second electron carrier here called FAD, flavin adenine dinucleotide. So it gets reduced from FAD to FADH2, which means we are oxidizing succinate to fumarate. We have a hydrolysis reaction where we add water to rearrange, and we have one final oxidation of malate to regenerate oxaloacetate, and we reduce our electron carrier to NADH, and now we're ready to take another acetyl coenzyme A in, bind it to oxaloacetate, and start the process. So with one acetyl coenzyme A coming in, we produce 2 carbon dioxides, 1, 2, 3 NADH energy molecules, 1 FADH2 energy molecule, and 1 ATP. We've gone through a ridiculous number of chemical reactions, and the cell is ahead by 3 ATP. If we did this citric acid cycle its second time for the second acetyl-CoA, we'd get 1 more ATP. So we'd have a total of 4 ATP, all 4 fingers, after glycolysis and the citric acid cycle. However, we also have a lot of reduced coenzymes, our other energy molecule. If we go back to glycolysis, we have 2 NADH that are made. We get 2 more from the transition step. That brings us to 4 NADH. If we do citric acid cycle twice, we get 6 NADH. So now we have 10 NADH chemical potential energy. And we have 2 FADH2. So our grand total is 10 NADH, 2 FADH2, and 4 ATP. The goal in the final step of cellular respiration is to do that electron transport. Remove the hydrogens from those reduced coenzymes and take them through electron transport that will generate a proton gradient. We can use that electrochemical energy to convert to chemical energy of ATP. This represents, in a tiny little cross-section, the mitochondrion. This is the inner mitochondrial membrane. Outside of that would be the smooth outer mitochondrial membrane, and there's that space between them. The blue blobs represent a series of electron transporters, and that shuttles the electrons from those electron carriers. The NADH enters at the first set of carriers. The electrons flow through, the protons get pushed into this intermembrane space. FADH2 enters at the second stage, and it also will pass its electrons and its protons. And at the end, our protons are pumped into this intermembrane space. The electrons keep flowing through our carriers until at the end, we combine electrons with some protons that remain in the matrix, and we reduce oxygen to water. We do end up with an excess of hydrogen ions, of protons in this intermembrane space. That does a few things. We have potential electrochemical energy now. We can't just move those protons by simple diffusion because they are charged. Charged particles do not freely cross membranes. It also changes the pH. If we have a buildup in excess of hydrogen ions, that will lower the pH. And that is why this electron transport is oriented in the way it is, so that this change of pH is happening in this intermembrane space where really nothing else happens except for this proton pumping. What do we do with those excess protons? Well, they are a proton gradient, an electrochemical gradient. And again, we have our inner mitochondrial membrane, the outer membrane, the intermembrane space where the hydrogen ions are building up. Now we have facilitated diffusion. This molecule is called an ATP synthase, an enzyme to help us synthesize ATP. And the energy, the electrochemical energy from the hydrogen ions moving through literally makes this enzyme spin. That creates mechanical energy. That mechanical energy will take phosphates and attach them to ADP to make ATP. So this is called oxidative phosphorylation because the energy from oxidation reactions drive the phosphorylation of phosphate plus ADP to make ATP. So we had calculated how many coenzymes, how many electron carriers had we generated from glycolysis, the transition step, and the citric acid cycle. We saw that they are going to be donating their electrons in electron transport. 1 NADH generates enough of a gradient to generate 3 ATP. We had 10 NADH, so 10 times 3 is 30 ATP. That's why it was important to keep track of those reduced electron carriers. They're carrying energy in a form that can be useful in electron transport. And the FADH2, because it enters later, generates enough of a gradient so that we get 2 ATP for each FADH2, and 2 times 2 is 4. So we get 34 ATP from electron transport, plus the 4 that we got from glycolysis and the bridge and the citric acid cycle, for a grand total of 38 ATP energy molecules from one glucose. And that's not even 100% efficient. There is some energy that will be lost as heat, but we get 38 energy molecules for one molecule of glucose that only had 24 atoms in it. So cellular respiration and the use of this electron transport chain is an extremely efficient and effective way to extract maximum energy from food molecules. In the next part that we will do after the exam, we're going to talk about anaerobic metabolism. The cellular respiration is aerobic respiration because oxygen, air, is the terminal or final electron acceptor. This is how we do metabolism. It's how anything that does aerobic cellular respiration does metabolism. Not all living things require oxygen. Some are capable of doing types of metabolism if they have to when oxygen is removed, and others can only carry out metabolism in the absence of oxygen. The respiration that we have covered is aerobic cellular respiration, and we will save our anaerobic types of metabolism for after exam 1.